releasing proteins from a cell suspension by means of sonification. Check if the room is occupied before you enter. The following materials are required for sonification: a pellet of bacterial culture, for example E. coli with GFP-UV, disruption buffer, for example tris HCl 10 millimolar at pH 8, pipettes, pipette tips, Eppendorf vials, ice, 70% ethanol, a beaker, and ear protection. Resuspend the bacterial pellet in disruption buffer. Use a 5 ml pipette for this. Then, store the bacterial suspension on ice. If the protocol prescribes this, now you can add lysozyme and PMSF. Turn on either one of the two devices. There is a Branson and an LA Biosystems Sonicator. In this video, we will use the Branson. Always wear hearing protection while using the Sonicator. Set the amplitude. This is at 40% for the Branson and 70% for the LA Biosystems. Set the time. This depends on the amount of bacteria you have. A good starting point is 30 seconds. Place the 30 ml tube with your bacterial suspension on ice under the probe of the Sonicator. Check that the probe is at least half a centimeter into the liquid. Make sure that the tube is not touching the Sonicator probe. Sonicate for 30 seconds, wait for 1 minute, then repeat the cycle. Take note, it looks like the bacterial suspension boils. The suspension should not foam or become too hot. Determine how much protein has been released. This is done in the following way. Pipette 20 microliters of the sample into an Eppendorf tube. Spin the Eppendorf tube in the centrifuge for 1 minute at 10,000 Gs. Measure the absorbance at 280 nanometers of the supernatant using the nanodrop. The disruption buffer is the blank. For the operation of the nanodrop, see the relevant video clip. Repeat the following steps. Sonicate for 30 seconds and wait for 1 minute, then sonicate for another 30 seconds. Pipette 20 microliters of sample into an Eppendorf tube and measure the protein concentration again. Repeat until there is no increase in the absorbance that can be measured at 280 nanometers. This means that all the cells have been lysed and all the protein has been released. Keep the samples on ice as much as possible during the procedure. Clean the Sonicator with 70% ethanol after use. Turn off the Sonicator. Centrifuge the sample according to the protocol for 10 minutes at 5000 G. After centrifuging, transfer the supernatant into a clean tube.